Alright, let's talk about the small intestines. It's the place where we do the majority of our absorption. We did a little bit in the stomach of some fats, a little bit, some vitamins, but really the star of the show is the small intestines, and then when we get to the large intestines, that'll be for water. So the functions of the small intestines is to do some chemical digestions, mostly through enzymes and nutrient absorption, and that's going to go to that mesenteric proper. There's three parts of the small intestines. There's that duodenum, jejunum, and ileum. Remember how I told you to remember them? DJ, ileum gets the party started. And here's our little rapper guy. And he says, if I was a DJ, my name would be Enzyme because I'm always breaking it down. Well, that's exactly what happens in the small intestines. We take those large structures like those sugars and break them down into glucose. And we take the nucleotides or nucleic acids and break them down into nucleotides. So the small intestines has a lot of surface area, and how it does that is not just by being a long tube, which it is, but it's got villi, which are these finger-like projections, and in the next one you'll actually see that those villi have villi on them. So the villi have villi, so those are microvilli. The large intestines is a circular fold, and you'll notice in the cadaver that they sort of fold on each other, and that's to move that chyme around slowly in a spiral pathway, and that's going to lead to it moving around a lot, and it's going to lead to more surface area through a process called segmentation, which we'll talk about soon. The villi itself, they're the finger-like projections, and if you look inside of them, you'll see that there's arteries and veins and lacteals. The arteries and veins are where we're going to move all of those structures into, like we're going to move sugars and amino acids, and fats will go into the lacteal system. And this area, the microvilli, is called a brush border. Brush border is where we're going to make the enzymes and sort of help, that's where the enzymes functions will be. And there's two types of motility that's occurring here. There's peristalsis, of course, because we're still moving from the mouth to the anus. We're going to call that peristalsis, just moving towards the large intestines and eventually to the anus. But then we have this thing called segmentation, and that's a little different, where what we're going to do is we're going to take this chyme and sort of mix it constantly, going from this one bolus to two, back to one, back to two, back to one, back to two, and it's designed to mix it as much as you can, and that's going to create more surface area. That's going to create more areas for those enzymes that are being produced in those brush borders to activate and break that food material down. So this is definitely one of those mechanical mechanisms that we're using. If we look at some more of accessory organs, we're going to see that we have the liver. It produces bile. It also does blood glucose levels. It breaks down lipids into fats. It helps with protein metabolism, stores vitamins, destroys and damages red blood cells, and removes toxins. The gallbladder is going to be a place that we're going to be able to secrete bile. We're going to hold bile in the gallbladder right over here, and every time you eat food, that bile will squirt a little bit of that bile onto that chyme, and that will help break the fat down and then eventually move it into the lacteals. The bile itself is going to be a yellowish-greenish fluid. If bile gets into the bloodstream, that's going to lead us to this thing called jaundice, where the skin turns sort of yellow. It also creates this bile salt and cholesterol, and these guys are going to help us move fats around and things like that. It also has a pigment, and that pigment leads us to our feces being the color it is. That's going to be called bilirubin, and that's just bile pigments with waste from the heme, and that kind of gets into our fecal material. The bile salts, it's designed for emulsification, and we'll see that picture soon. Can't forget the pancreas. The pancreas is so important to making all these enzymes that without it, we wouldn't be able to do some digestion. People that get their pancreas removed because there's a tumor or cancer that grows in it has to take a lot of pills to supplement all of these enzymes so they can still eat what they need to. If it's really bad, they'll end up just eating a special diet, which is almost like a paste that they can just absorb nutrients without any of these enzymes. So it acts as an endocrine gland and an exocrine gland, because the endocrine is going to be producing hormones. The exocrine is going to go ahead through a duct, release those enzymes into the large intestines. Every day it does about 150 mils of that pancreatic juices, which really is a mixture of this water, sodium bicarbonate to bring the pH back down to 7, because remember how we're at 2 to 4, and then as soon as we start squirting into the large intestines, small intestines, we want to bring that pH back up, and that's going to be done through sodium bicarbonate. And then of course we have a whole bunch of enzymes, and refer back to your enzyme chart that we put together, and you'll see that a lot of them are produced here. The pancreas also goes ahead and makes some hormones. It makes insulin and glucagon. Insulin is going to lower blood sugar levels in the blood. So you have a lot of sugar in your blood, it's going to take that sugar and sequester it to the liver. Glucagon is going to be activated when your blood level is too low. So your blood is too low of sugar, and it's going to go to the liver, release some of that sugar out, and it's going to let you burn it. So glucose be gone from the liver, and insulin is going to insulate that sugar into the liver. The pancreatic juices, besides that, we talked about the bicarbonates because they're going to go ahead and create a perfect pH for the enzymes to work, and then the digestive enzymes that, again, go ahead and refer back to your chart to see what those are. Large intestines, they're going to be designed for water absorption. They're going to help with feces to sort of become solidified. This is going to be anything that the body can't use, but we're going to use these things called hostrum, that's these sort of folds, and they're going to help aid with the use of a lot of bacteria to go ahead and suck up that water and eventually make it go into a solid material. The divisions of the large intestines, we've kind of gone through them already in the cadaver, but we have the cecum, which is the first part, and what's attached to the cecum is the appendix. What's between the cecum and the small intestine is the ileocecal sphincter. Remember, the last part of the small intestines is called the ileum, and this part is going to be called the cecum, so ileocecal sphincter. The appendix is good for immune function. It has beneficial bacteria there. There's a lot of nerve endings there. So when it starts to expand, your body knows about it because of the pain. The colon is where we're going to have divisions, the ascending colon, transverse colon, and descending colon, eventually going to the sigmoidal colon. And then finally we have the rectum, which is going to contain fecal material. And then there, we're going to have two sphincters. We have an involuntary one that opens up, and when it gets full, it lets you know. And then when you find an appropriate place and time, you open up the voluntary one, and that's going to release all of that fecal material. The carbohydrate digestion we'll do in another video.